So hello and welcome. My name is Jocelyn Park. I'm the Marketing and Communications Manager at CTSI, and I'm here to welcome you to this month's Translational Science Seminar, Mentored Career Development in Translational Science. Penn State Clinical and Translational Science Institute's TSS series promotes research across Penn State University, the Clinical and Translational Science Awards Consortium, and throughout this community. This month, we are highlighting our Mentored Career Development KL2 program, led by co-directors Dr. Sciamanna and Dr. Lorah Dorn. We will also have the opportunity to hear from two former KL2 scholars, Dr. Steffany J. Fredman and Dr. Steven Hicks, who will share how the program created opportunities to advance both their research and careers in translational science. If you have any questions during the presentation, please feel free to drop them in the chat and we'll capture them for the Q&A portion of this seminar. And without further ado, I'd like to pass off to Chris and Lorah if we're ready to do so.
Jocelyn, also I did see something pop up in the, I guess the chat box, but I can't access it. Steve Hicks had a question, if you can check for that. And then I don't see Chris yet, and he was going to start. So
right. Yep, let me see. We have here. Not sure what's happening. I cannot see what's in the chat. Steve here.
Someone else has also raised their hand. Hong.
Yep. Right. There we go. I got Steve in.
And Steffany, would you mind sending it to Chris again? And maybe which Chris email did you have? He has a Penn State Health and a Gmail.
I replied to his email.
Oh, you did. Okay, that should do it.
Zoom link in.
So maybe send it again, I guess.
Hey, can everyone hear me?
We hear you, Steve. We don't see you.
Yeah, when I try to click start video, it's saying you cannot start your video because the host has stopped it.
Okay. You should be able to now.
Oh, yes.
There we go.
Hi, Steve.
Hey, everyone.
Good to
Sorry
see
for
you.
the technical difficulties.
That's okay.
And no Chris yet that you see, Jocelyn?
No.
Okay, well, if you can put up his slides, I'll
All
start
right.
with his stuff.
Go ahead. Can you see my slides okay?
Yeah, I see your welcome slide.
All right. And next.
So the next one, okay,
Oops, sorry.
good. So first of all, we're thrilled to have people in the audience, even though I can't see who's in the audience. So that's always a little bit of a challenge, but we're very glad you're here and excited about telling you about the KL2 program and also highlighting two of our past scholars. And so this just is a slide from, we refer to it as NCATS, the National Center for Advancing Translational Science. And it gives you a nice picture, I think, of telling how people do talk about translational science. I think most frequently, people think of bench, the basic science circle on the right, to bedside, although it involves much more than that. But if you can keep your handle on that, I think that will be helpful. So all of our scholars do research within this realm, some of them across domains, and I think you'll hear some nice examples of that. And the next slide, Jocelyn. Okay, so let me tell you a little bit about the KL2. It's a 2 to 3 year award, and what the award is 75% of your protected time in your current position. And these are usually assistant professors on campus, both Hershey and University Park. And then there's about $25,000 a year for their research projects. And this KL2 is like an institutional K training award. In NIH terms, the K can be an individual K, where people write a grant to be funded for 5 years of training and research, or an institutional K. So the KL2 scholars are on an institutional K. It's designed as the goal to prepare junior faculty to gain an R01 award or equivalent. And an R01, for those of you who don't know, is one of the larger NIH awards to an individual to conduct research. In our program, we include a weekly grant writing seminar. That actually is something new that we're starting this year. We also have monthly didactic seminars on a variety of topics. The KL2 program has a curriculum with different content areas that we focus on that have to do with translational science, and also have to do with being a successful translational scientist and leader. So for example, we have different trainings, not only on grant writing and publications, but we might have some things on leadership. We have things on regulatory science, so scholars understand that. Of course, things about team science. How do you develop your own lab? Even things like crucial conversations. How can you talk with a boss? How do you talk with your team members across all those realms? So it has a variety of things beyond what the scholar themselves proposes for their science and for their training. They meet with mentors frequently, and also have the opportunity to go to the national meeting to present their translational work. We can do the next slide, Jocelyn. Okay, I want to tell you just briefly about our current scholars. We have 8 of them listed in alphabetical order here. Sunny Bai is in the College of Health and Human Development. She's an assistant professor. I'm just going to read these because I don't know how, or Chris, there you are. Would you like to get started with this? I just started with Sunny, but I'll let you continue if you'd like, or I can.
Better late than never, yes. So we're excited for the scholars are from UPark, 4 from College of Medicine. So Liza Behrens is going to do a pilot testing dignity. She even came up with a clever acronym for this too as well. So decision making in nursing homes. Lauren Forrest is doing a project on personalized assessment and prediction. I could read these, and I will. Short-term suicide risk for people living in rural areas. Emily Homan, dynamic predictors of obesogenic eating behaviors in children. We've got a health services research project with Sarah Horvath, impact of Pennsylvania Medicaid policy change in preventing pregnancy. And then Lena Huerta Science, early initiation of anti-inflammatory meals to promote partial remission in type 1 diabetes in youth. Elena Pierce, neurocognitive and behavioral factors that promote resiliency to pediatric obesity in rural communities. And lastly, but not leastly, Susan Valdier will be doing a project improving cardiovascular health behaviors with gardening, development of a scalable mobile M health intervention. So a lot went into these getting to this point. So we are really excited to get going. And even though we know that it's really just the beginning, The goal is to help launch these 8 people towards independent research funding down the road. But we want to get to the main event too. So,
2 of our really great scholars from last round. So, and Jocelyn, I believe you're introducing them. Is that right?
Yep.
Oh, good.
Go ahead and stop sharing. And Steffany, I believe Steffany is going first if you want to share your slides, and we'll see if you have any difficulties there.
And Jocelyn, if I could just share a couple things about the 2 scholars, is that all right?
Absolutely.
Okay, great. I wanted to highlight for you because I'm not sure that everybody on knows about translational research and translational science. And earlier I gave the example of bench to bedside. So I think these 2 people really illustrate that very well, and in 2 very different fields. For example, Steve, to me, does the bench to bedside that most people would get. He gets biomarkers and he puts them into clinical practice, among other things, developing biomarkers and that. Steffany, on the other hand, I think does the same thing, but it's surprising to many people that in behavioral or social sciences, you can do the equivalent of bench to bedside. And I think from Steffany's, you'll hear how great that example is, and I won't spoil the punchline. So thank you. And you're on mute, Steffany.
I wasn't sure if Jocelyn was going to say anything else about my background.
No, go ahead. You're fine to go.
Okay, great. Thanks. Hi, everybody. I'm Steffany Fredman. I'm an associate professor of human development and family studies and psychology here at University Park. I'm a clinical psychologist by training, and I study the intersection between individual mental health, particularly post-traumatic stress disorder, and couple and family functioning. And so I do both basic research, looking at how PTSD can disrupt couple and family functioning, as well as how what's going on in the individual's couple and family life can facilitate or impede recovery from the disorder. And as Lorah mentioned, I also do translational research in the area of couple and family-based interventions for PTSD that are designed to simultaneously treat the disorder and enhance intimate relationship functioning. So before I get into too much into the weeds of my talk, I wanted to make sure to acknowledge that it really does take a village to do this kind of work. So team science, it also takes money. So I wanted to thank my collaborators and the CTSI for investing in me and this line of inquiry. So this is a picture of my KL2 collaborators who are from Penn State, University of North Carolina, Chapel Hill, Heidelberg University, and Stanford University. It also includes my students. And so we were all convened. This picture was from May 2018, several months into my KL2, where we had convened for a 1.5 day long gathering that was supported with funds from the KL2, as well as an early career professorship I had gotten from the College of Health and Human Development, where we could really do a deep dive into the proposed work from the KL2, as well as think about future projects that would come from the KL2 And also I'll tell you at the end too, how not only have that seeded current and future work that I'm doing, but also current and future work that my students are doing, which has been really exciting. So as I mentioned, I study the intimate relationship context of PTSD. So PTSD is a major public health problem. It affects at least 8% of the US in people's lifetimes. Rates are 2 to 3 times that in military and veteran populations. PTSD is associated with major psychosocial impairment, accelerated aging, and even premature death. It costs the US economy billions of dollars annually as a result of lost productivity, as well as healthcare costs associated with the disorder and its physical and psychiatric comorbidities. And importantly, the effects of PTSD are not contained to the trauma survivor. The effects of untreated PTSD ripple outward to afflict the broader family system, particularly romantic relationship partners and children who are themselves at elevated risk of psychological distress and physiological wear and tear as a result of the stress of living with someone with chronic untreated PTSD. So there's a lot going on in this figure, but let me just kind of put it up there as a conceptual illustration of what my colleagues and I have pulled together to try to understand factors that are happening within and between romantic partners that can maintain the PTSD as well as associated relationship impairments. And in particular, what I want to highlight is that there are well-documented associations between PTSD and romantic relationship problems, particularly in the area of conflict management and intimacy. And those in turn give rise to relationship discord, physical and psychological aggression, and partner psychological distress. What's less well known is why. So what is happening within and between these partners on not just kind of over time, which is what this slide is really depicting, the research that's shown like over periods of months to years. We know these different constructs are associated, but what we really don't know is what is happening on a momentary basis that could account for these problems with conflict management and intimacy that contribute to this host of other problems. And, you know, as I and my colleagues started thinking about it and reflecting on this, what probably links all of these problems together is how couples relate to, for each person in the couple, how they relate to their own and their partner's emotional arousal during times when strong emotions are likely to be experienced and expressed. And there had been some research, like in the couple years before I applied for the KL2, there was growing research using this voice stress paradigm to study couples. And then in particular, some of my colleagues who are collaborating on the KL2 were using this paradigm to understand what was happening on a micro time scale from an emotion regulatory process in couples where one person had individual psychopathology. So for example, obsessive compulsive disorder or eating disorders. And so there was some interesting work looking at how emotional arousal communicated through the voice could predict disorder-specific patterns of responding, both to one's own emotional arousal, as well as emotional arousal that was signaled through the other person's voice. So let me just very quickly explain what this is. So basically, if you were to decompose the speech sound wave, the fundamental frequency, which is referred to as F0, is one of the most information-rich and robust signals of emotional arousal. And so basically what it's reflecting is how fast the vocal cords are vibrating. And so when the person is more emotionally aroused, the vocal cords are vibrating more quickly, there's more tension in the voice. And then as the air is from the lungs is passing over the vocal cords, that's producing sound, and that's heard by others as pitch. And so higher pitch in this way can be a window into people having more emotional arousal. And this is really important because vocal fundamental frequency serves an important communicative function. It signals, typically it can signal emotional distress. It can also just signal high levels of emotional arousal in general. And in couples, the emotional arousal of the two partners often become interconnected or related to each other, at where one person's emotional arousal is influencing the other person's emotional arousal, either in adaptive or in maladaptive ways. And so what I started wondering when I thought about what I knew about the individual psychopathology of PTSD, where emotional arousal itself can be a trauma cue, and people go to great lengths to avoid feeling their own emotional arousal, I started wondering, well, is the reason that they get so dysregulated during conflict with their partners, and why they also tend to also get very dysregulated and withdraw from efforts at closeness with their partners, is because the emotional arousal that they're experiencing internally, or that they're picking up through what's communicated in their partner's speech, is that essentially triggering them? Is that making them, even on some kind of almost unconscious level, feel like in some way they're kind of being reminded of their trauma? And my colleague, Dr. Amy Marshall, who was one of the mentors on the KL2 grant, actually had the perfect archival data set to explore this question. So what the primary research study as part of the KL2 was looking at how trauma survivors and their partners regulate emotional arousal on a moment-by-moment basis while they're communicating, specifically during two different conversations, one that was designed to elicit conflict and one that was designed to elicit emotional closeness or intimacy. And then to test out this idea that they're responding to their own emotional arousal, consistent with emotional arousal serving as a trauma cue, we were looking at how specific PTSD symptoms would predict these patterns of emotional responding. So, spoiler alert, this is exactly what we found. But let me tell you how we got there and why it'll be relevant to you as part of your KL2. So this was pretty complex, and it required new training for me. So I needed to learn new methods for studying short timescale processes in couples with PTSD. On the assessment side, needed to understand how to mine this vocally encoded emotional arousal, so the fundamental frequency. And then once we had that, how to analyze that using dyadic longitudinal data analytic techniques. I also needed more experience in grant writing. And so that was accomplished by training and mining of the F0, regular meetings with my mentoring team, sitting in on Neelam Ram's intensive longitudinal data analysis class, attending multiple quantitatively oriented seminars that focus on intensive longitudinal data analysis. I myself presented sometimes to get feedback. I went to a week-long workshop at the University of Utah focused on dynamical systems modeling. I workshopped grant ideas, and then I also submitted grants. And I wouldn't have had time to do this if I hadn't had the protected time and the additional financial resources from the KL2. So just very briefly, the technique we were using was the repeated measures actor-partner interdependence model. So after mining the voice stress, and actually here in the right-hand corner is data from an actual couple in the study. And so what you can see is we're mining time series data. And then what we're trying to look at is the pattern of responding. How are people responding to their own arousal? So did where they were the previous talk term predict where they were going, as well as was where their partner was at the previous talk turn, did that predict where they were going? And then we wanted to see how that would be predicted by these specific symptoms. And so what we found was indeed people were responding to their own emotional arousal, which is what we would expect if there's some consistency. But what was really cool, and as we had hypothesized, and what people in general would be intuitive, but nobody had shown this, was that people who were walking around easily triggered by their trauma reminders are more reactive to their own emotional arousal in these emotionally intense situations, and then they get stuck. So where they were makes it much more likely that they're going to keep going in the same direction. So if you are being emotionally aroused, especially if you're very angry, it's going to be harder to change gears and calm down. What was also really cool was that tendency also gets sort of counteracted by avoidance. So this is what we see in PTSD, is people are constantly being triggered by trauma memories, and then they try to avoid so that they can escape that arousal. And what we saw here was that this is not just playing out over periods of weeks to months to years, but we can literally see this playing out on a moment-to-moment basis while they're interacting with their partners. So then when we looked at the patterns in a totally different conversation, so they're also in the lab interacting with their partners, but now they're told, okay, instead of discussing something that's an issue of concern between you, we want you to talk about a positive issue, and they picked the topic. What we found was not only were they responding to their own emotional arousal in the same way that they were in the conflict discussion, where they get more stuck, the higher level of, like, the more easily triggered they are in general, but they're also responding to their partner's arousal as if the partner's emotional arousal is also serving as a trauma cue. So they're not only being set off by what they're experiencing internally, they're also getting set off by what they're hearing in their partner's voice. And similarly, those who tend to try to avoid were less responsive to their own arousal and their partner's arousal. So what you're seeing is across contexts, they're responding in a very similar way. So not only are they getting easily triggered by their own arousal and their partner's arousal, but then you see on this micro time scale, they're trying to escape that arousal. Interestingly, we also found that if your partner was kind of more emotionally detached, that also made it harder to downregulate from your own arousal. And this, while not hypothesized, actually made complete sense to us and might explain why emotional numbing symptoms in one partner are the symptom cluster that's most strongly associated with why partners are less happy in relationships. Like, essentially what this is showing us on a micro time scale is that people have to work harder to regulate. I have to work harder to regulate my own arousal if I'm partnered with somebody who's walking around disconnected from their emotions. So to summarize, I know that was a lot. Basically, this showed what we had hypothesized, that during both conversations, trauma survivors are responding to their own emotional arousal in a manner that's consistent with arousal serving as a trauma cue. During the intimacy discussions, not only are they responding to their own emotional arousal as if it's a trauma cue, they're responding to their partner's arousal in the same way. And this also is a micro timescale window into why partners might be more distressed when the trauma survivor is more emotionally numb. So this finding these were very necessary and important results to be able to use as preliminary studies to then apply for grants building on this paradigm. So we're continuing to explore these emotion regulatory processes using other voice speech samples from the same archival data set. I'm also now including this paradigm in both pending and future grant submissions that involve clinical trials to test couple-based treatments for PTSD. So as a result of doing couple-based treatment for PTSD, are we shifting these patterns? And are these treatments potentially targeting this mechanistic process that's maintaining both the disorder and relational difficulties? We also have a planned R1 submission, a new submission for next March for a special call that almost feels tailor-made for this project, where they want basic research trying to understand dyadic psychosocial processes and how they relate to health outcomes. And so we're really excited to be able to use the voice stress paradigm as one level to analyze these processes. We'll also be bringing in biosignals from wearables, and ecological momentary assessment and observational coding. And I just also kind of wanted to end that not only did the investment in my research program with this paradigm and this training help advance my own research agenda, but it's also helped my students and my mentees. So my graduate student, Annie Lay, was able to, under my direction, complete a dissertation using daily diary data. So it's not quite as micro as what we were doing with the voice stress stuff. So it's one level up, looking at daily diary, but published 2 papers in the top family science Journal of Family Psychology, to understand daily relational processes between couples, members of a couple who have transitioned to parenthood, which is a very high stress time. My recently graduated grad student, August Jenkins, had an F31 funded by the National Institute of Minority Health and Health Disparities, looking at using daily diary stuff to understand the linkages between individual mental health and couple functioning in Black couples. And the first paper from her dissertation has been invited for a special issue at Journal of Psychopathology and Clinical Science. And then Emily Taverna, who is one of Amy Marshall's grad students, used this paradigm and same sample, but different voice recordings as part of a successfully funded NSF graduate research fellowship and her psychology minor project, which has just recently been submitted for publication. So with that, I'm going to close. But if anybody has any questions, please feel free to reach out to me. And I'm also happy to talk with anybody offline if you have questions.
Thank you, Steffany. That was wonderful. Now we're going to introduce Dr. Steven Hicks. Let me know if you have issues sharing your screen.
All right. Can everybody see that okay? Great. I'm going to move my camera just a bit here. So, hi everyone. My name's Steve, and I'm a general pediatrician here in Pediatrics, and I'm also a physician scientist. And today I'm going to talk to you a little bit about the project that I did as part of my KL2 training. My talk is going to be a little bit different than Dr. Fredman's was, partly because I don't think my research is nearly as interesting as hers, but also because I think it might be useful to maybe put my project in the context of lessons learned and how the next generation of KL2 scholars can maybe apply some of those lessons in their own work. So what I'm going to do today is discuss all the different ways that mentorship and peer feedback and protected research time can be leveraged by KL2 scholars to catalyze their career, and then share some of my own insights from my project, which involved multi-omic biomarkers for child brain development. Specifically, my project was looking at whether we could identify specific molecules in saliva that could help us to diagnose children with autism. And the rationale for that is that currently there's about an 18-month wait for a definitive diagnosis of autism. And so the average age of autism diagnosis in the US is right around 4 years old. But we know that the earlier that we can make a diagnosis, the better neurologic outcomes that child has. And the main reason that diagnoses get delayed are because kids wait on really long wait lists to see developmental specialists who have to use semi-subjective tools that take a long time to get trained to use and then a long time to actually administer. So in thinking about kind of the essence of the KL2 program and what its goals are, I thought back to my own application and how my overarching goal was to become an independent researcher. And I suspect that many of you included some sort of similar phrase on your own application. And then I got thinking about, what does it mean exactly to be independent? How do we define that? Is it because you went on to get your own grant funding without a mentor? Is it because you have a certain number of senior author publications? Or because you have your own research project that you developed all by yourself and nobody else helped you? Or is it that you just no longer really need a mentor? And so it's kind of like, I feel like this meme here exemplifies this conundrum a little bit, right? It's hard to know sometimes when you're flying by yourself and you're sort of dependent on somebody else's wings. So first, mentorship, right? You've all entered this process with assigned mentors. You also are going to have excellent mentors through the KL2 program and Dr. Dorn and Dr. Sciamanna. But it's important to know kind of the qualities that you want in a mentor, because these shouldn't be your only mentors. You're going to work with people on a daily basis that may not be official mentors for you, but you can always learn something from them. And so the kind of people that you want to have as mentors are honest people that are going to give you honest, critical feedback. They need to be knowledgeable in your subject area. They may not need to understand the complete ins and outs of your project, but bring something to the table that it pushes your knowledge in a specific content area beyond where it currently exists.
Steve,
yeah.
Sorry for interrupting. I don't know if we aren't seeing your slides advance. I'm assuming you have advanced.
Yeah. Okay.
Great.
Sorry about
Thank you. That's okay.
So let me see.
We see just the title slide. And it actually was not in full screen mode or whatever it's called.
Thanks for telling me that.
Yeah. Thanks, Laura.
Sure.
I could also pull them up if you want to tell me.
Yeah, do you mind doing that?
Yeah, I'll go ahead and do that.
I'm struggling to get them to pop here.
There's that good picture you were talking about, Steve.
Now it makes a lot more sense, huh?
Yes.
Okay. Great. So Jocelyn, are you going to advance them then, or will I be able to click on this? I guess you can do it.
Oh, you just tell me when to advance. Are we on the next one?
Yeah, so this is essentially what I'm going to talk about today is like these four things, right? So how do we identify or define an independent researcher, and how do you know when you've made that leap? So yeah, next slide. And so the first thing I'm going to talk about is mentorship and how you pick a good mentor and then make sure you utilize it in the best way possible. So I just spoke a little bit about what I think are some of the important traits that you look for in mentors. And also thinking about the fact that a mentor is not this very clean box of mentor or not mentor, right? Every person that you interact with in your research on a day-to-day basis may bring something to the table that you can learn from. So we're constantly receiving mentorship. And I actually don't think that mentorship ends even when you become an independent researcher. I don't think that's the way that we should identify or define an independent research career. Now that I have an R01 and I'm graduated from the KL2 program, I still have people that I use as mentors, maybe not in official capacities anymore, but I feel like mentorship is a constant thing. It's a two-way street. While you're in the KL2 program, it's important to come to your mentorship meetings prepared. You should have SMART goals, so measurable goals that you can understand when you've achieved them. You should be ready to accept critical feedback from your mentor, and then really nurture the relationship. It's not your mentor's job to reach out to you and let you know, hey, we haven't met in a while. That's really your job as a KL2 scholar. And so I think taking that on as a role or a job is important. Next slide, please. So along that line, I would encourage you to use your peers in the KL2 program as mentors. So for me, that person was Dr. Chandrika Gowda, who's in pediatric hemonc. Our projects were similar in that they involved a decent amount of molecular biologic science and involved pediatrics, but really the overlap ended there. But that did not stop us from being able to give each other what I think, hopefully for her too, was valuable feedback. And the way that we did that was on the drive back and forth from State College. So she and I would carpool every day. Every month when we drove up there. It's 100 miles, an hour and 45 minutes. And it was really good time to just decompress from what we had just learned in the KL2 seminars for that day. I don't know if we're still doing that, traveling, and Dr. Dorn, is that something we're still doing?
For the monthly meetings, yes, we are. And we've added some, because for exactly why you're saying, we think it's really important for you all to get to know each other and have that opportunity to talk. So I'm glad you brought that up, Steve.
Yeah, so I know that some people have maybe Can you guys hear me?
Now I can.
Okay.
Yeah.
I know that some people have complained about that as a downside to the program, but I really saw it as a very positive, a thing that you could turn into a very positive experience. So I would encourage the next group of scholars to find people to ride with and to use them as peer mentors. Okay, next slide. All right, so next, are you an independent researcher because you've developed your own project? I know probably everybody who's in the incoming class KL2 scholars has a project that they worked with a mentor to develop in some way. So although it's their project, they may not feel complete ownership over it. But I would say that I think good science and team science are one and the same. And it's unlikely that you're going to move on from the KL2 program and develop a project in a vacuum all by yourself. In order to get externally funded large grants, you have to be prepared to work with collaborators and maybe share the credit for the projects that you're developing. So for me, in my work with molecular biomarkers, whether that's for autism or concussion or other pediatric conditions, the team that I have generally on every project includes at least one medical expert other than myself. Even though I'm a physician and I see patients with concussion or autism, I try to find medical content expert to be on the project with me. And the same goes for molecular experts. So I understand biomarkers and how to measure them, but having somebody that does this on a full-time basis, I think is really important for me. Same with statistics. So a big part of my KL2 training was learning how to do the statistics on the projects that I do. And I feel really confident now being able to look at a large data set and analyze it. But at the same time, I know that that's not always the most valuable use of my time. And so I still rely on statisticians to do that work. And then just having a regulatory coordinator and a research coordinator and a lab tech that keep things humming along kind of behind the scenes. And one of the really important things I learned from the KL2 program was how to first identify those people to hire in those positions, and then also kind of keep them motivated and all rowing in the same direction towards a larger goal. And so I'm sure that you'll learn those lessons as well. And I'd encourage you to pay close attention to them because I found them to continue to be really valuable in my work. Next slide. So next, I just want to talk about your research question for your KL2 project and how you kind of can think about building upon that. So my aim of my research project as a KL2 trainee was to develop biomarkers in saliva that would help identify children with autism. And you can see in those first two papers there on the left that we basically did that right off the bat in 2 papers. But so the aims are settled, they're satisfied, we've completed the project successfully. But every time you answer a question, you should kind of stumble upon 2 or 3 new ones. And it's important for you to chase down those loose ends while you're in your KL2 and you have the protected time. Make use of that time, because those kind of hanging strings that you're going to follow are going to be what leads you to your next big grant submission. So for me, in developing biomarkers for children with autism, one of the kind of secondary questions that came up was, what factors are affecting these biomarkers? Do they change over time? Are there external factors we need to control for? And so in the top right-hand corner, you'll see that paper in PLOS One, diurnal oscillations. Basically, that's a lot of big words to mean that the molecules that we measure in saliva, they change throughout the course of a 24-hour day. So they have circadian rhythm to them. And if you don't control for that, you won't be developing accurate biomarkers. So I actually took this secondary question and turned it into an entire R01 submission all by itself. And so that's an example, I think, of how a secondary question can turn into a really interesting idea. And then the same thing goes for the Frontiers in Psychiatry paper in the middle bottom of the slide. So just like Steffany mentioned, for her project, having like this tailor-made R01, the NICHD put out a question or an R01 call for papers or projects investigating biomarkers of gastrointestinal dysfunction in children with autism. And so we had this data set ready to go, and that R01 went in just 2 months ago, and it'll get hopefully discussed in June. So here's an example of like exhausting every possible question surrounding your research topic. Next slide. But I'd also encourage you to think about how you can take your research topic and then turn around and apply it in maybe a whole different area you never thought about before. And so for me, that was concussions. And I used what I had learned about biomarker development and molecular measures and longitudinal clinical trial design to develop a whole second area involving biomarkers for detecting concussion and then predicting who would recover from concussion in a short time versus have prolonged symptoms. And this is what ended up being my first R01 funded study out of my KL2. So we are now in our, I think, just started our second year of this project, where we are looking at adolescent children ages 13 to 18 with concussion and trying to predict who will be recovered in 4 weeks and who will not, so that those children can hopefully be triaged into treatment programs immediately following their injury, instead of just taking a wait and see sort of approach. All right, next slide. So another way to perhaps measure whether you've become an independent researcher is by senior author publications. You may or may not have noticed it because the text was so small, but in 2 slides with all those papers, my name slowly migrated from the first author to the last author. And I think that's an indication that you're the one driving the work, identifying the question, and then mentoring others to write up the data. So that is one way to measure independence, but I don't think it's the only way. And I don't think you should sit around waiting for others to write your papers for you so that you can be a last author. That's not the way to move forward in your career. I'd also encourage you to shoot for the stars on what you think are your best results, because if you're not getting rejected, then you probably aren't submitting to high enough impact factor journals. And lastly, I would just say that cranking out papers is the way that you are going to impress reviewers when you go to submit your R01. And so you really need to take advantage of the protected research time you're gaining in the KL2 and use it to submit as much as possible. All right, next slide, please. And that takes me to grant funding, which I think is probably the best way to define when you've made it as an independent researcher. Most of us would probably agree that the R01 is kind of the gold standard for this. But I would encourage you to also think about lots of other funding mechanisms, right? There's lots of ways to get your projects funded, and maybe your project doesn't fit well with an R01. Maybe it's better suited for NSF, for the Department of Defense, or a specific foundation. And those are all ways to get your time protected and have your own independent lines of funding. One of the realizations that I came to about halfway through my KL2 training was that only about 10 to 15% of R01 grants are funded. So if you do the math, that means you're going to have to submit somewhere between 6 to 8 R01s before you get your first one, unless you think that you have somehow a better project than everybody else out there. So doing the math, you have three years maybe in your KL2. That's not a lot of time to submit 8 R01s. So I would be thinking ahead about what those R01s could be. And I would not wait until you're in your last 6 months to start writing them. For me, I have an R01 in concussion, but I also have an R42 and an R61. So I looked at lots of different mechanisms. And to get those 3 grants, I submitted, I think, 11 or 12 grants to the NIH. So no better than anybody else. But just submitting a lot is really, I think, the key to moving on to an independent career. All right, next. So with that, I will wrap it up. And just like Steffany, I'm happy to answer questions via email or stop by my lab in the Crescent building at 7754. And thanks for your attention.
Thank you. Great, wonderful presentations, everyone. We do have a question for Steve in the chat from Lena. She also noted a great presentation, and she asked, did you ever apply for a K08 or K10 before submitting your R01?
I did, yeah. So I submitted a K23 actually, and it got scored, I think, mid to low 40s. And the research strategy got scored really well, and the training strategy was what got trashed. And in carpooling with Dr. Gowda back and forth, she said, she was the one who actually encouraged me and said, you know, maybe they're criticizing your training because you don't need any more training, and you just switch to R01s. And so that's what I did. And that worked out well for me. But I know that's my specific experience, and everybody's going to be different. I think it's definitely logical for some people out there to be submitting K08s or K23s at the same time you're in the KL2.
Any other questions at all from folks? Steve, I really like the point about having everybody you bump into can be a mentor. And you really have to look at that. Everybody can, bring some different kind of advice, and it's amazing. You build, kind of a Rolodex, to use an old-fashioned term, of people you can kind of ask for all different aspects. And most people are more than happy to do that.
Yeah, couldn't agree more.
And also the fact that both of you, I mean, it was so impressive that you both wisely used your time, knowing it was not easy, but you did exactly the right thing. And we can sure see your successes, not just from today, but all the other things that we can see on paper too. So thank you for sharing that. It was just super.
Steffany, is Google going to be listening now to my calls to tell me what's wrong with my mental health? They're already, I think, can screen me for Parkinson's, right? I think that's pretty well proven. That works well in voice. It might be just the next piece of data we're emitting all the time.
Yeah, the DoD is investing a lot of money to try to look for a vocal biomarker of PTSD. And people are also looking at that for depression too. I just wanted to say, again, to thank the CTSI, thank the KL2, thank you, Lorah and Chris. You've both, I would say, both been part of my mentoring team over the years, and I've had these conversations that have been incredibly helpful, both for grant submissions and also professional development. And also, as Steve said, the other members of the KL2 provided invaluable feedback, both on my research and also with the professional development. So it's just an incredibly rich opportunity, and I'm so grateful for it. So thank you.
Good to hear.
Same here.
We did have one last question in the chat from D. Any changes to what KL2 brings?
I'm not sure that's something you can kind of go through in a minute.
Yeah.
So unless, Lorah, you have a sense of what Dee's asking that can be quickly encapsulated.
Actually, not quite. I'd need a little clarification. Sorry. If Dee's still there.
I'll make one comment quickly. Steve, you'd be proud then, because we're hiring, there's a new kind of administrator that's going to be helping with the KL2. And on her first day of work is our first day in UPark, and I am picking her up and we are driving together, sort of, talk about, what'll happen with the program, et cetera. It's a lot of time. You can use that time to really, dig deep into conversations, and it's very rare you get that kind of time with one human being. So I totally agree. You use it for what it's worth.
Yeah, they have that van, right, that you can like ride in and get your work done because it has internet. But honestly, like, I just felt like I could do that anytime in my office, like by myself. I'd rather just like connect with somebody and get some good one-on-one feedback. So yeah.
Great.
Awesome. Yeah. Anything
Great.
else, or are we good? Jocelyn, you're running the show, right?
Kind of. I mean, you guys are the stars of the show. So we are at just past 1. So I want to thank Lorah, Steffany, Steve, and Chris for your presentations today. To our participants, thank you for joining. We will be sending out a follow-up email with the links to the recording and the slides so you can dive in a little bit deeper. So with that, I hope everyone has a great weekend, and thank you for joining this month's Translational Science Seminar.
Thank you, Jocelyn.
Thanks, Jocelyn.
Thanks,
And
Jocelyn.
Steve and
Bye, everyone.
Steffany.
Thanks, Evan.
Bye, everybody.
Bye.